Um, so I'm a new assistant professor in Department of Pathology, and it's great to be here because most of my work are bioinformatics actually. So our lab do spatial system biology, understanding how cells organize, localize, form neighborhoods, and talk to each other. And specifically, we're focusing on neurological disorders spanning from both psychiatric disorders and also traumatic brain injury. So today I'm inviting you human brain and mouse brain to see what spatial omics can help us understand the complexity of the brain. To start with, I like to talk about this oil painting and the state, and each break are visualizing the identity and the demographics within a map. So here you are showing, I'm showing the real estate median housing price by zip code. And you should appreciate that the median housing price are not just affected by the house build itself and the neighbors. It's the same as tissues. So it doesn't matter which cell type you are, your function not just depending on where you are located and who your neighbors are. Taking brain as an example, so the particular very interested in is the hippocampus, where it's about a 4 centimeter long region here highlighted in cyan. So this one is the center of your memory, emotion, and mood regulations. We started with hippocampus, but the spatial omics technology can actually apply to any regions of the brain. The reason we're focusing on hippocampus is because there's a lot of neural anatomy established in former studies. And a same excitatory neuron, depending on the location in hippocampus, can have very different functions. Hippocampus has a region-specific trisynaptic circuit. So the dentate gyrus, which is the pink here, is the commanding center. And the green region here is CA4 region, CA3, CA2, and CA1 region. These are the messenger region. So the signal will usually come from the entorhinal cortex, which is here, going to dentate gyrus, the commanding center, going processed and going out to the CA3, and then transmit to CA1, and later going to subiculum. And subiculum will project neuron projection to cortex, fornix, or other regions of the brain. So it really depends on where the location of a neuron is. The function can be really different. It can be a signal relay, or it can be a memory formation, or it can be a synaptic regulation. To understand such a complex composition of cell types, there's an emerging technology called single-cell transcriptomics, and most of you are pretty familiar with it. But I like to critique that the challenges in single-cell studies is that there are no spatial information. And if you recall, the very first step of single-cell study is that you need to dissociate the tissue. So during that mechanic or enzymatic dissociation, you're losing a lot of the subcellular transcriptomics profile. For example, in a neuron, the neuron body itself, the nuclei itself is probably just 10 micron, but the axon and the dendrites can be millimeters away from the cell body. So during enzymatic dissociation, majority of the mRNA in the neuron processes, in the dendrites and in the axons will be lost. And secondly, when people are dissociating the cells and process it, the cell morphology will also change. So you will lose the in situ morphology and cell-cell organization in the resulting data. And most importantly, there will be no spatial landscape or mapping for how cells interact with each other and how tissue organize together. So the two challenges from single cell to spatial is that how can we actually barcode the tissue with a spatial index? How can you deliver such a small index to label the location of the mRNAs? And the second biggest challenge is how can you work on such a small number of molecules? So a single cell usually have only 2 picogram of the RNA, where traditional bulk RNA-seq are working with 10 nanograms of RNA. So these are 5,000 times difference in the total RNA you can collect. when you're only having 2 picograms of the RNA, like you have to consider the noise, specificity, the bias in amplification during PCR, and a lot of other things. So spatial transcriptomics actually spearheaded 2 mainstream, one from imaging-based method and one from sequencing-based method. So from imaging-based method, I organized a chart adapted from so starting from the very upstream for spatial omics, depending on if you want mapping of the entire tissue or not, if you don't, you only selected the whole region for laser, you can go with nanostream. Other than that, if you want to specific regions all show up in the same map, then you either go with imaging-based method or sequencing-based method. So imaging-based method, the resolution is super high. It's like optical resolution, 200 nanometer usually. But the downside is that you only look at what you're looking for. So you need to design the probes for specific transcripts or specific protein. For sequencing method, the beauty is that it's very unbiased and systematic. So you got to look at all of the transcriptome or all of the open genome. However, the downside is that how do you actually barcode the tissue? So there might be gaps and there will be very big spot size when you're targeting a specific library. So the resolution is usually about 10 to 50 micron, where to 200 nanometer, which is subcellular resolution. But depending on exact what biology you're trying to study, there are many other factors kicks in when you're choosing a spatial transcriptomics or spatial proteomics technologies. And this slide is also in my lab website resource page. So for people who want to look at it again, it's publicly available online. So giving this technology available in mind, then how can we actually use it to study the human brain? So this is the project I spearheaded at Columbia University, where we established a spatial multi-omics pipeline to study the human brain, the hippocampus, and we collect 17 human brains, 9 neurotypical and 8 MDD. So all of the patients are sudden deaths, meaning there's either cardiac arrest, gunshots, stab wounds, or industrial accident. So there's very short axonal state. There's no prolonged life support. So there's no hypoxia. There's no changes in the brain. And we also did thorough toxicology and autopsy report. So we know what's the cause of the death and what are the substance used in the brain, in the blood, and in the hair for heavy metal and also for substances. So we collect the fresh frozen brains in sequential slices. Usually we collect around 40 slices at the same time. And then we use one for spatial proteomics, one for spatial transcriptomics, and one for spatial epigenome sequencing. The whole idea is that how can we build an integrated map of the same brain? So for people who are interested in the cohort, this is the very first time we are releasing the cause of death. So most of the other brain sets are usually in human cortex or neurodegenerative disease, but there's a huge need in how can you include neurotypical brain in the study, and how can you provide the complexity of confounding factors such as gender, metabolic state, toxicology, and other underlying disease. So this is our data set. It's a very clean reference data set for both neurotypical and psychiatric disease. So these are all treatment naive. So although they got not diagnosed with major depressive disorder, but they did not take any medical treatment. This is the slide I got asked most. So I made a video of it. How do we actually prepare tissues for a human brain? So we collect a coronal section of 2 centimeter thick frozen human brain. And then a hippocampus is embedded in the temporal lobe, which is the red square here. So it's very small, only about 2 sections of the coronal section will contain the temporal lobe with hippocampus. And we're focusing on the mid hippocampus labeled by the lateral geniculate, where the optic optical nerve are crossing. So we try to compare the same plane across all of the different brains. And then we have dry eyes on the bottom of a big foam box. And then we put the tissue block. It's like a rock. So we need to have a dental saw to identify the specific region and then to cut it. And when you are pretty close to crank it open, we stop it, and then we take out the tissue block about 2 by 2 centimeter mounted option and cut it at 10 micron thickness. So this one does not have any embedding OCT. So the beauty of the brain is that it's very homogeneous. Most of 80% of the brain are lipid. So when you are freezing it, they can freeze pretty homogeneously. But imagine if you're working with pancreas, bone marrow, or other tissue that have different state of like a calcified region or fluid or other different densities, it will be very challenging. So no OCT embedding, and then have very fresh, frozen, unfixed brain here. We just put the slide onto a regular gas slide. And then the question we ask is that, can we build a single cell Google map of who's there in the hippocampus? So with spatial transcriptomics, the biggest criticism we have for it is that you don't have cell morphology and you cannot see the ECM. Those are just transcript detected with 50 micron for Visium or 2 micron for Visium HD. But for protein profiling, you can actually see the cells and you can see the functional protein. So this technology is called multiplexing proteomics code detection by indexing. It's invented by Dr. Gary Nolan's lab at Stanford University. It's really a bioengineered innovation where they're only using primary antibodies conjugated with a DNA barcode. And then you incubated all of the antibodies with the tissue slide overnight. And then we put it onto the imager here with microfluidic chambers and vacuum chambers. And then it has a fluid system to wash and to go over. So every cycle, we have a complementary DNA barcode will bind to the primary barcode with fluorophore. So every cycle, we only image 3 channels. And because this binding and unbinding are mild, so we can strip it off and rehybridize another different 3 cycles of the fluorophores. So usually one experiment for 30 markers take about 4.5 days. The machine itself is automatic, but there's a lot of troubleshooting when we're using the engineering like trial model. The beauty of this is that it overcomes the traditional IHC. So instead of just having 4 antibody at the same time, we can look at as many antibody as possible. So the current limit, it's about 40 antibodies. The limit is mostly financial decision, not scientific decision, because one slice like this is centimeter scale. It costs about $6,000. And the difficult part is that one antibody works, but you have to have all antibody work at the same time. You need to do timely or regularly titration of the antibody activity, and you need to validate their shelf life and concentration. So we spearheaded the panel design of the protein in the brain. And then this is what the raw data looks like. So it's thousands of images for each channel stitched together. And then this video shows it's a stack of high resolution images. You can toggle on and off for different layers. Let me play it again. So for instance, the first layer for DAPI is all of the neuron, all of the nuclei, and the cyan for just the neuron, and then neurofilament, yellow here for axons, and then PSD 95 for all of the dendrites. The beauty of it is that you can look at all of the protein, one experiment, all of the protein at the same time, and you can magnify it up to 300 nanometer, and you can screen through like a wandering in the universe, who is there in the hippocampus and who are their neighbors. And you can see even at the magnified region, there's no diffractions, there's no overlaid artifacts. And this is a blood vessel identified. For people who are studying BBB or drug delivery, they will be their primary target. And we can also look at single channel for collagen, for vimentin, or for Claudin 5 for the endothelial cells and also the tight junction in the BBB. But hippocampus have a lot of neuroanatomy. Just so we have very high quality of imaging data doesn't mean that we can quantitatively analyze it and compare between different cells or different samples. So the next biggest question we have is that how can we create an expression matrix of the protein data? So as you see here, hippocampus is highly organized in different layers. Just the dentate gyrus region here, which is in cyan, has 3 layers, which has the axon-rich region, the dendrites-rich region, and also the neuron-rich region. And this bottom yellow, the have blood vessels. And pathologists actually have a lot of insights for what cell types they are just based on 6 markers. For instance, in the granular cell layer, you have cells like granule. They are very round, very densely packed, and their nuclei are big comparing to their cell body. Where in the conno-ammonis region, the neurons are like a pyramid, like a triangle. So just by looking at the morphology and the size, we can give them identity. An oligodendrocyte can be marked by OLIG2, which is their unique nuclei stain. And so with this like domain knowledge, we naively thought it would be very straightforward, but it actually takes 3 postdocs to convert the deck of images to a tabular data showing here. We want to acknowledge Dr. Yuki Tan from Gary Nolan's group at Stanford, and we invented a new multiplex imaging analysis pipeline called Spaces to analyze this type of data, codex data. So what we essentially do is that you use the DAPI plane as the cell mask for the nuclei, and then it collects all of the fluorescence intensity of that mask for different protein channel and create the tabular data. So each row here are representing one single cell, and each column here are representing one protein expression. And we also have the X and Y location of that specific cell. So once you have that, you can do high dimensionality clustering, treating it as a protein expression matrix, and then can like toggle on and off for different cell layer in a vector map and change the size of how the vector is and see how they are correlated. So looking at the analysis here, so we designed a panel of 25 targets and can image one single slide of hippocampus at the data you're showing here represent 82,009 cells in one whole human coronal section of the hippocampus. And we created a pipeline to generate a tabular expression matrix where you can treat protein data as expression data and project them into n-dimensional space and visualize by UMAP. So they all of the neurons still cluster together here, and all of the blood cells and immune cells still cluster on the top. They're more similar. And we can, because we have their XY location, so we can just call specific cell types once we identify them in the unsupervised clustering. What's more powerful is that traditional IHC, when you have the imaging, there's a big problem of background, noise, battery effect, antibody activity, and many other things, and tissue artifact. But with our pipeline and data set, we are able to integrate 16 human hippocampus together. So you could see here the cell are actually clustered by their identity, not by their sample source. So the sample source are actually mingled together, but the cluster are actually by specific what protein expression they have and how different they are from each other. And what's more interesting, because it's spatial data, so we can calculate how cells, what is the distance between each other. So we've defined 200 micron as the largest cell-cell distance of one cell to another, and then calculate the cell pairs. What is the average distance of a cell to another cells? So in total, we examine millions of cell pairs, and then just for your interest, like we find that in MDD patients, the microglia to microglia distance is much shorter, meaning they aggregate together, so they're staying closer to each other. And this is not just a few pairs picking from the images. These are unbiasedly profiling of the entire 1.6 million data set. And in the future, we are thinking if you have a diseased tissue and one cell is like microglia are activated, And you could tell the cell-cell distance to see there's an immune region here, there's a vascular region there, because cells are stay closer together, they're activated, then they will be in different state. And what's more exciting is that this type of single cell map of the cells from the functional level, and we're not just understanding the cell at the cell type, we are trying to understand how they organize and how they form neighborhood, and how do neighborhood correlate with each other or interfere with each other, and ultimately how do they organize as a tissue type. So the idea is that cells are not independent. They work as a community. This is human intestine using spatial proteomics identified from John Hickey's paper in the Nature Human Atlas project. So what they have is the human intestine cell type identified codex data, and then they run the neighborhood analysis. It's like a rolling window of 200 micron, keep rolling for different regions, and then they will group the regions with similar cell types composition. So amazingly, their method working for intestine also work for the brain. So we use the same algorithm. And if you look at on the top left here, these are just individual cells colored by their cell type. But if we want to compare the same conserved region across different samples, we need to know what's the unique neighborhood. So we run this neighborhood and identify very conserved neighborhood. For instance, the pink here representing the granular cell layer in dental gyrus, and the yellow here, the big band, is the cornel ammonis region, the pyramidal neuron region in the cornel ammonis. And you can visualize the composition of each neighborhood by different cell types. So the beauty is really like when you're scaling up, when you have so many other data and you don't actually know a lot of neuroanatomy, this is a very powerful way to identify conserved neighborhood in different samples. So without even knowing anything about hippocampus, you could identify the cornel ammonis marked by the yellow here and the dental gyrus marked by the pink here. So in the future, this type of study can be used to understand if there's a specific neighborhood in your tissue or in a disease, or say if you're surrounded by 10 CD4, CD8 cells and 1 macrophage, then that specific neighborhood means that you're in trouble. So this is really taking the power of the spatial, the location, into play for interpreting the cell function. And what's more, the spatial proteomics can also be integrated with single-cell RNA-seq. That's so exciting. As I just said, cells are not independent. They are very dependent. And genes are also not independent. They also depend on each other. So this state-of-art algorithm developed by Dr. Zhongming Ma's group at UPenn and now at Yale. So he assumed that if you have RNA data and also protein data, how do you build the relationship of these two weakly linked omic modality? And the idea is that because gene 1 and gene 2, they are dependent, so likely they will stay closer in the same cell type. For example, GFAP and vimatin in astrocyte will always have the relationship in the same RNA testing for all of the astrocyte. But in the protein, the GFAP and vimatin will also have that relationship. So preserving those internal relationship, they do a cluster first and try to match the cluster in RNA and protein of the same cell type iteratively. And then they give a best prediction for, hey, this cell's RNA is most likely to present in that protein expression because that's by prediction. So using our spatial proteomics data, we also examine 1.6 million single-cell hippocampus single nuclear RNA-seq data, which they identify 68 subtypes that we project their cell abundance in the spatial data. So in the future, like if you have reference single-cell data and you want to know where they're located in a protein imaging data, multiplexed, there will be this is the best method we experienced so far. But without going too far for spatial omics, I'm very greedy. I also want to cover some epigenomics. But to wrap up the first part, we created a very comprehensive triomic map of the human hippocampus that that encompassing the proteomics, the mRNA, and also the open chromatin, which I'm going to go deeper in the next part. So this will be the very first atlas of the human hippocampus result from different modalities. And it can serve both as a reference and also as a training data for new algorithm of cell-cell distance, of cell neighborhood, of gene regulatory, and also cross-modality relationship. In our experience, we have 25 protein in the proteomics, and we examine those exact protein mRNAs in the spatial RNA data. So only about 30% of the time, they are correlated. So majority of the time, the protein and RNA, even they're representing the same function protein, they're not correlated. So I guess this is very interesting for the field to know that in the future, if you have the RNA, you should know that their protein are probably not following the same trend. Only about 30 to 35% of the time, they're correlated. And next, I want to shift gear beyond proteomics and transcriptomics, which is epigenome. And I know people here are, I see a lot of experts working on long-read-seq, working on DNA methylation. But here, the technology we're focusing is chromatin accessibility. So spatial ATAC-seq is a method I helped develop when I'm a PhD student at Yale. So the idea for spatial epigenome sequencing is that we want to understand what is the opening, what is the gene regulation of the tissue in a region-specific manner. So if you are a cell, what would be your cell memory? Like, how can we read what experience of the cell has from their epigenome? So for instance, if I'm a cell, I'll remember what my type is, what is my fate is. So that's my lineage. So it can be read from like tissue or cell type specific marker in their epigenome. And think about all of your somatic cells. You have the same set of DNA. The reason you have different cell types is because you have different regions of the epigenome that are going to be regulating which genes get transcribed and which genes doesn't. But all of your somatic cells are having the same set of blueprint. So using spatial ATAC-seq is really very critical to understand what is regulating the gene expression. So we're going one layer up, like not just looking at what's changing at the functional level at protein or what's changing at the gene expression at high throughput, but also what is regulating, what is regionally regulating. So the model we're using is traumatic brain injury. The rationale behind is that for psychiatric disease, neurodevelopmental disease, those are not a strict spatially variant disease. But traumatic brain injury is that you can control the regions of the injury, the impact of injury, and the duration of the injury. So in human, there are three typical types of TBI, which is blunt injury, like a blunt force exerted on the brain, and also a rotational injury, like motorcyclists, like they have to have a very quick acceleration. The brain doesn't touch anything, but there's a contusion of the brain and the skull. And there are also blast injury, like shockwave blast in the battlefield. And depending on where when you're looking at the injury, the process happen in the brain can be very different, like from minutes to days to weeks, like different processes are dominating in the brain. So in the very acute phase, which is the first injury, that's usually the most golden therapeutic window. Gliosis happens and also acute inflammation happens. And then following days to weeks, people will usually experience chronic inflammation, axonal injury, and also neuron death or neurodegeneration after mood changes, or they have a personality changes, or they have like other functional changes. To model this, we use a state-of-art fluid percussion injury, where it's a whole mechanic setup. So where you have a hammer on the side and you can adjust the angle of the hammer, and there are multiple sensors for pressure, and you can measure the force. So in the center here, this is a fluid chamber filled with liquid. And then when the hammer hits the fluid chamber, there will be fluid moving from one side to the other. And at the other side, we will mount the fluid chamber onto a lure open in the cranial window. So we remove the skull of a very small 2 mm skull, removing it from a neuroectomy. And then we mount the lure on top of the brain. So the injury is not on the skull, but the injury is on the brain. So the actual injury happened in milliseconds. So we took this off the switch and the hammer will fall down, and then the liquid will quickly insert it like a membrane on the fluid would touch the brain. So the brain is like a layer with 3 dura. So the fluid will cover like a very even coverage among the 3 dura matter. So this is like an overlook that we're using traumatic brain injury as a model to understand how spatially variant the RNA, the epigenome, and the proteome. So for the sake of time, I'll just jump to our conclusions. So the idea is that we want to establish an unbiased platform to understand what is actually changing after TBI and how can we make better decisions for treatment, because every human have a different response after the initial injury. Some people are more likely to have edema, herniation, clogging, or bleeding after the initial injury, but some people don't. And the region of the secondary injuries really cannot be predicted. So here we are introducing spatial multi-omics to profile which regions specifically are being changed after TBI, mild TBI, and what cell types are involved, and what is regulating those changes. So we have 4 samples sequenced. One is the control sample, which is here, and then 3 TBI samples. And among the 3 TBI samples, these 2 are from the peri injury. So if you have an injury center, then there are also injury plane, a few 300 millimeter across the anterior or posterior of the center. So the idea is that not just the center of the injury plane you will have changed, also the side, the peri side will also have it in coronal sections. And the last TBI sample is the most severe one, is the injury center, where you can already see the force have a difference. So the different number of the genes are expressed. So the injury side is on the right. So right-hand side, so you could see here, just for gene expression number, they're not very different in the first 3, but in the injury center one, a lot of the cells are stopping their gene expression and much less gene can be detected along the trajectory of the injury. And what's so interesting is that if without knowing anything about TBI, without processing or integrating anything, if we just look at traditional markers for TBI, we could already appreciate that these markers can represent what's going on in the brain. So for instance, for gliosis, which is a marker, we're using a marker GFAP to mark for the astrocyte. You could see here in the control sample, they are very symmetric, while in the TBI brain, it got a lot of astrocyte in the hippocampus, in the fiber tracts, and also in the meninges. So those are the firefighter, the first responders in the brain that are going to go and detect what's the invaders, what's the change. And then we also detect different neuron activity for ion exchange or for neuron transmitter transporting. So you could see here the function of the neuron actually being shutting down at the injury site, where in the TBI samples, both for glutamate transporter and ion exchange here are like dampened. Well, in the control side, they're not. They're like very symmetric. And my student, Roy, which is on the first row here, intelligently clustered the cells based on their UMAP. So we want to identify not just by visualize, we want to do high dimensionality analysis and understand the annotation and the region and the change over time. So he clustered the spots using a SORAT and then made this video where you could see here each color are representing a key region of the brain, where the light blue are representing isocortex, the yellow here are representing ventricular system, the subventricular and the lateral system, and the purple here are thalamus, hypothalamus, so their neuromodulation, sensing, and then feeling and touching, and then also have striatum here in the red region, so for movement, initiation, modulation, and stoppment, and also amygdala for fear. So if we just do an unsupervised clustering, we just see the region are clustered together. But what's more interesting is that how can we understand the function? So the region, if you cluster them, they will cluster by their specific anatomic region. But we really want to understand what is the relationship between different genes. So we use Giotto to identify what are the patterns of gene correlation. And using this one, we can find very specific gene pathways got turned on for neuronal activity dampened at the injured site, for a very elevated stress and immune response at the isocortex, amygdala, and striatum for movement, for fear, and for rationale. And gliosis are mostly in hippocampus and at the meninges. And there's also some change at neuromodulation. So the somatosensory region in the cortex are also activated. And we can run like heat map to see how different genes are co-clustered together. But what's like going back to the talk I just like prime you, how can we understand the gene regulation here, knowing that there's a change in their gene module and pathways, and they have a different spatial heterogeneity? How can we understand the spatial epigenome? So the method we're using is called spatial ATAC-seq, and I bring actual labs, the chip setup here where audience can stay and take a look at the actual chip. So the idea is straightforward. So when the genes are being transcribed or not transcribed, it's mostly because their regulatory region are open or closed in a condensed state or in an open state. So using TN5 transposes, we can cut for the open region that are not bind with a histone. And this is a multi-step mini reaction, mini chemical chips. So the beauty of our spatial barcoding chip is that we can confine where the biochemistry reaction is happening, and then we can realize multi-step reactions for cutting, for adding a barcode, for adding another barcode, and for in situ reverse transcription. So you can amplify it first, and then you take it all at the same time. But because they are already barcoded with spatial barcodes, so we can just resolve computationally after we sequence. So the idea is very different from spatial transcriptomics, where people are designing probes on top of the Visium slide. You're capturing all of the poly A tail. But here we're not barcoding the probe, we are sending the probe into the tissue. So the experiment starts with a tissue slide on the bottom and the PDMS slab made by soft lithography. It's a technique in electrical engineering where you can use light to etch specific small channels at your resolution. So here you're seeing like 50 parallel microchannels at 10 micron in resolution, but the resolution can be variable depending on your need. And then we like manually load each spatial barcode at this side and apply vacuum at the other side. So this small spatial will flow in, become suction, and then you will incubate with the tissue to be added to their open chromatin. And this is how the barcoding works. So the reason DB6 is very powerful is because you can have different mini reactions with the tissue instead of like Visium or Xenium. These are one-step reaction. You only have a functionalized surface with your tissue. So there's no further like complex biochemistry can be made. But our data, for the very first time, we're resolving what is happening after traumatic brain injuries. That here you are seeing each dot are representing 50 micron sequencing library, and each library are containing the open chromatin short DNA peaks detected by sequencing. And then because of the size of the chip, we can only detect half of the hemisphere. So we use two adjacent slices. One, we detect the non-injury side. The other one, we detect the injured side. And then we put a data object together and analyze it in the same data object. So you could see here, like it's not as clear as RNA-seq for brain anatomy, but it's telling you a lot of brain anatomy as well, where the red here is thalamus for somatosensory. The yellowish green here are hippocampus, the neuron-enriched region, where the purple region here are the isocortex. So what's more interesting is that we identified like the top 100 different motifs. So these are detected by ChromaVar, where we're calculating the motif score based on their chromatin opening. The color here are representing how open the motif can be detected. So if we can detect red here, it means that ATF3 can bind to those motifs. So those motifs are in open state. But if it's blue or it's light yellow here, it means that those motifs are in condensed state. The ATF3 cannot bind. So looking at this motif score, you can look at how responsive the tissue are for specific transcription factor. And specifically, we find the ones most interesting are the AP1 family, which are the master regulator in post-traumatic brain injury. And ATF3 is just one of the stress responder. So you see it's like selectively activated only at the injured side. And Force and June are other family members in this AP1. So I told my students that I think of them as godfather because the transcription factor function as a family with hierarchy, and then they exert their force regionally. They're like each region has their different dominating forces. So instead of looking at all of the non-coding region, we start with the transcription factor motif region that we can know what is regulating the downstream genes. And for reference, we also look at CRAB3, which is another transcription factor, but do not have the asymmetric distribution that much as ATF3. And NFIC is like a myelin regulator, like transcription regulator for myelin cells. So it doesn't have a preferential distribution at the injured site. And we also look at the downstream genes in their RNA profile. These two are target genes by ATF3, so there's our immune response genes. So the work is still ongoing. My lab just got started here like in February. So we are looking forward to collect more transcriptomics, proteomics, and also epigenome data to understand how the brain works and who's there, how they get regulated, and how they get organized. And I like to open the field a little more that single cell and spatial analysis actually cannot just be applied to traumatic brain injury, but it can also be in many other topics like development, neurodegeneration, psychiatric disorder, drug abuse, and neuroimmune disease like muscular sclerosis and substance use. I felt we're at the age where Picasso are painting the cubism for the same guitar player. So the two paintings are portraiting the exact same player, but the information can be interpreted very differently. We felt that we're still in the age of the cubism, the simplicity to an extreme. All the information, it is there. The question is that, can we approach it? And with that, I'd like to thank all my collaborators at UMichigan. Dr. Thomas Sanderson Lab provide the traumatic brain injury animal model. Dr. Josh Welsh Lab provide a consulting for data procurement and analysis. And the first part of the talk for human brain data are in collaboration with Dr. Rongfan's lab, Dr. Yan Xiangden's lab at UPenn, and also a lot of colleagues at Columbia Psychiatry, where they are maintaining and collecting a big fresh frozen human brain data and tissues. And my lab is hiring and recruiting at all levels. And I'd like to stop here and take questions.
Thank you. That's a beautiful presentation and amazing slides. Given your multiple collaborations, maybe this question is a little more complex than I thought, but I'm interested of the beautiful things you've shown us. Which of these findings are new, not previously reported by anybody, and which ones are setting new directions for this field?
Yeah, wonderful question. So what new findings can we get from spatial omics? For human, it remains a big technical barrier. How can you provide? So we are the first group establishing the workflow, how you can profile fresh frozen human brain in the hippocampus region. So there's no other like reference data available. There's single cell data, but there's no spatial data for human hippocampus. It's just technically they are degraded very fast, and people cannot get that region in big coronal section, or the region can be very small. So there's no other omics. So first, we provide the very first reference. And second, we're the very first group examining what happens in neuropsychiatric disease. I did not show the finding here, but what we find is that if we do cell type deconvolution for hippocampus and we can compare between disease versus neurotypical, we actually find that in healthy, the cell distribution pattern changed. So in oligodendrocyte, they co-localize altogether. But in inhibitory neuron, in the healthy, they are mutually exclusive. They are governing each specific region. But in the MDD patients, their inhibitory neuron are not functioning. They don't have the right feedback. And their RNA velocity are also changed. It's just for the sake of time, I did not get chance to present, but it's not just the expression, but it's also the dynamics. And it also the cell-cell distance and cell organization that are changed. But depression is a very subtle disease that there's no even ground truth. Psychiatry itself is a very subjective the degree of the disease. There's no even quantitative metrics like a blood test or imaging to decide who has depression or who doesn't. So this is the very first study that we can learn what happened in their brain, what are their gene changes, what are their gene regulation changes. Yeah, so for now, like we're setting up the first step, what is changing in their epigenome and how the cell are differentially organized in the MDD. And for TBI, like I think we have more leading advantage that we can resolve the real-time difference in TBI in the entire same brain and identify the top different pathways at different time points. So we can clinically, we can provide them with a lot of insights, like do you protect the neuron in the first 3 hours, or do you protect the immune response in the first 3 hours? People actually don't know. Specifically in mild traumatic brain injury, if you look at the histology here, the cell density are the same. Their distribution are also very similar. And even for the neuroimaging with MRI or CT, they're not changing. So there's no way to ask a fresh human to donate their brain to study what's changed after TBI. So this is providing a very unbiased and systematic way to understand what's actually happening, what will be the best function of therapeutics after the initial injury in the golden therapeutic window. I hope that answered the question.
No, it's wonderful. And I'll just give you one quick perspective. In the 1970s, I spent several years where I spent 4 a.m., 5 a.m. in the morning at the morgue to collect brains to try to do something along these lines.
Mm. Yeah, the Michigan. The difficult part for human brain study is that you cannot predict when they pass away or when they will, which relative will sign the consent and donor agreement. And when you want to have very fresh brain, so these are all post-mortem interval less than 24 hours. There has to be 2 teams standby, 1 social team, religion team, and 1 medical team. So before the relative sign the do not resuscitate form, those team have to reach out and get standby and transform. So 4 hours is critical. So if the person died within 4 hours and there's no 4-degree or cold storage is exerted, the tissue will degrade very fast and you will not have spatial proteomics or spatial transcriptomics data. Yeah, so I have to be on standby mode, on call like 24/7 in my postdoc years, and I'm only 800 steps away from my lab, so I'm in ready for the 5 years. And here, the Michigan Brain Bank, the Columbia, they have their private collection. Here, the Michigan Brain Bank, they have 4 full-time working on just the autopsy and the tissue transport and the consents and medical record coordination. Just having a brain doesn't mean you can study it. You need about 6 months to collect the medical record and clearance, and another 3 to 6 months to perform the toxicology and talk to pathologists, physician, and then their medical examiner or the coroner to collect all of the data together. So not one person have all the data. It's a diplomatic training as well. You have to talk to many people.
I thank you. I, like Dr. Omenn, when I did the Visible Human project, I spent a lot of time doing this kind of stuff too. And for the major depression, I would encourage you to look at the anterior cingulate cortex, where there's documented disruption of neural circuitry as well as likely changes in the multi-omics spatial distribution that could be very informative. I'd love to talk with you about that. That's a target for deep brain stimulation and treatment-resistant depression. And it's a known place where there's actually a disruption. And it could be adding a nice part of your story. It's a little bit different region, but I think it's something that you would find very interesting and happy to talk to you about.
Absolutely. We want to image the whole brain, not just the hippocampus. So you are talking about anterior cingulate, which we're hoping to do, but it's a financial decision mostly. That hippocampus has the right anatomy and right size to start with. Yeah.
Very good target.
I think so. Great talks. I have 2 questions. One is how much you can tell about the different types of neurons in hippocampus?
In proteomics, it depends on what markers you have. In proteomics, we cannot. So because we are not imaging specific neuron subtypes, that's the limitation of markers. So we're only using 25 markers. But once we project it with the single cell data have about 25 subtypes of neuron and 13 inhibitory neuron and 12 excitatory neuron. So we can see specific. I do see that question coming, so I have a slide for it. Hold on. So we can see the neuron subtype by projecting single cell data to proteomics data. So this is one way. And the other way is that we have spatial RNA. We can do cell-to location, like cell type deconvolution to predict the single cell resolution. But both of them require single cell reference data to see the subtype. The proteomics itself cannot. Yeah, it's just the marker.
Thank you. Our second question is related to Dr. Athi's question. I just wonder why you chose the hippocampus, because you said some budget things, but say it's relatively small, and let's say if you look at say sensory cortex, like in visual cortex, it might be easier. So how did you guys choose the hippocampus?
We choose hippocampus because of first we get MDD patient and like the brain, and the hippocampus is the center of the limbic system for mood disorders and regulation and spatial memory. So we thought hippocampus would be the very cornerstone to start with. But when you talk about a sensory cortex, it's a big region similar to cortex. You're probably talking about 20 times larger in volume than hippocampus. And everyone can have slightly different gyra and sulci. How do you control the exact same plane? But hippocampus, the mid-hippocampus can be identified by lateral geniculate, where you can actually define the specific region of all of the subjects. So it's actually the starting point. But in the future, what our Human Brain Initiative are trying to do is that looking at all of the brain region, not just hippocampus, but striatum, basal ganglia, amygdala. Yeah, amygdala for fear. Very popular amygdala.
Right.
And yeah, most of the study are looking at prefrontal cortex in neurodegeneration. But our department, the reason I joined pathology is our department handle forensics, autopsy, and also tissue growthing of the brain. So I got to actually go and touch the actual brain, and we have very good training for neuroanatomy. When you get this solid brain, like you need to resolve. Just looking at this, can you actually tell which is a motor neuron and which is somatosensory? It's actually a very vague area defined. So we actually, we like better neuroanatomy defined region to start with.
Cool. Thank you so much.
Thank you. Great talk, Dr. Xiao. So this is perhaps a more technically oriented question. So I read in the SpaceC manuscript, there's great novelty in the first part of your talk with regards to, the amount of spatial characterization that we're able to perform. But my question then as a follow-up is how accurate are those spatial characterization techniques? So what I mean by this is that for cell-to-cell interaction, it'd be easy to go into like QPath software and just look whether or not cell-to-cells are really like approximately close or repulsing each other. But for these larger order structures like cellular neighborhoods, neighborhood spatial contexts, right? We know there's like different methods for like functionally characterizing spatial proteomic data from IMCR tools, SquidPy, yada, yada. And so I was curious to hear your thoughts, like experimentally, how would you validate this? Or do we have to reconcile the fact that there's things that are not obvious to the human eye? So like, how do you look at like an image and experimentally validate like, oh, this part of it is like cellular neighborhood 1, and it's interacting with cellular neighborhoods 4 and 6, for example.
So basically you're asking how do we know our observations are true and what are the ground truths of pathology?
Exactly.
So hippocampus is a very well-defined region, and there's a book about hippocampus. So there are 6, hippocampus is archicortex, not neocortex. So it usually takes about 5 years clinical training in neuropathology to identify different regions of the brain and different cell layers of the brain. So there's a lot of ground truths for us to validate each marker and validate each layer. So for hippocampus, it's actually easy. But for nuclei, like in the midbrain, like red nuclei, substantia nigra, or basal ganglia, those are the challenging part because it's 3D, and depending on where you cut, there's no reference plane. It's very difficult. So I guess the field is still progressing to build the reference as a collaborative approach. But the Human Brain Initiative has been ongoing. It's like the next genome project for 5 years already, and we still cannot sequence the entire one single slice of the coronal brain. One human coronal brain is like 6 by 8 centimeter. Imagine how can you actually coordinate all of the transcriptome, epigenome, and proteomics in that such a large slice. You need to customize your glass, customize the surface chemistry of the glass, and then develop entire new coding strategy or library construction strategy. So I felt the field is still involving like better resolution, larger capture area, and more modalities. But I don't have a best answer for that. I think we need people like you to join.
Okay. Thank you. And just final question. So for the accuracy and like sensitivity of like detecting cellular neighborhoods or tissue domains, are there any like benchmarks with comparison to like other spatial proteomic methods?
No, we did not. So this is like we are just trial because they have the code from Gary Nolan's lab and say, okay, let's try and see how it works. But there are many methods for cellular neighborhood, and we are very ready to learn and try it. So if people have better ideas for how to define, here we are just using cell identity, but we did not use any protein expression. So ideally, we can use all of the information. If people have better idea for cell neighborhood and neighborhood interaction or correlation, we are very open for collaboration.
Yeah, I think that's of great interest. Is like, is a cellular neighborhood detected by one method conserved across other methods, or is it just arbitrary, like Delaunay triangulation or something like that?
Yeah, I felt so far this is the method that work for our data, but we just try 3 neighborhood detection analysis, and every method is specifically designed for different type of data. So it's not just for protein. So I guess we are still learning, and will give you a better idea.
Thank you.
One more question.
Yeah, that's kind of like a perfect segue into this, because I noticed that you said one of your findings was that in MDD, the microglia were closer to each other, right?
Yes.
And so it made me think like, well, does that mean because they're clustered at sites of where they think there's injury and that kind of thing? And then I guess my question is really, it kind of led me to think, it's about the neighborhoods, but it's really the pathologist, right? Initially, I thought, oh, well, maybe a pathologist can see this. But then I realized, no, the pathologist can't see all these cell types, right? So do you think like basically, I mean, what it seems like is you could study the whole thing as a network now with different nodes being different cell types. And then can you go into 3 dimensions? I guess that's really kind of my question.
We are ready to learn what you propose approach for that. Yeah, that would be super powerful. There's a way to do like hydrogel embedded thick section, like 300 micron thick or more thick. Then you can actually scan through the entire tissue section in the brain. It's called from Dr. Xiao Wang's group from Broad Institute. So they can use hydrogel to expand the tissue section, and then they can have a 3D reconstruction. But that's the best technology I heard of. But still, they have very small capture area, like 1 by 1 millimeter in region. So something like human brain is probably too big for them and too difficult to image. But I felt that's the mainstream. So people are doing bigger region and more from 2D to 3D and building a reference how we can register back to the coordinates in the reference.
Oh, that's great. Yeah, and I think on the 2D side, I know Naoki and I and Aditya from my lab, we're going to be meeting soon to think about how to maybe use geometry, you know, just like for your neighborhood kind of analysis. So maybe there's something that'll come out of that.
Yeah, that would be
Yeah.
wonderful. So for pathologists, this is what we pathologists do. So we do one specific stain. This one is doing nasal stain. So pathologists look at the stain for TP43 or tau protein or myeloid protein or just general neuron, and they identify one modality at a time. So it's impossible to look cells all at the same time. So I felt digital pathology is catching up with spatial omics and provide a more comprehensive study. Thank
Thank
you.
you so much for Dr. Xiao's wonderful talk. But I want to remind everyone, Dr. Xiao is the new members of the CCMB and also shareholder John McPalmer's in the DCMB. We can see there's a lot of data and information behind the scenes, how the multi-omics may interplays or crosstalk in the spatial. So manners, there's a lot of topics we can study together. So we look forward to interact more often with Dr. Xiao in the future. Thanks, Dr. Xiao's wonderful talk again.
Thank you.